So, well, hello, I'm Melina. I work at the AI Virtanen Institute, and I'm doing the PhD at the University of Eastern Finland. And today I will share with you what I've been doing so far. Just for you to know, this is a part of a broader project including different modalities, but I will stay with my part today. So let's break this title into the three key parts, which are the data that we have, rat brain histological images. What we want to do is to cluster or to classify tissue from those images, and the main method we propose are autoencoders. So here we have an histological rat brain section of 30 micrometers thick that has been stained for myelin. So myelin surrounds most of the axons in the brain. So by staining myelin, we are able to infer the location and distribution of the myelinated axons. So if we look closer, for example, corpus callosum or internal capsule, we have areas that are highly myelinated and that represent really different axonal distribution. We have more areas like cortex, dentae or amygdala, in which the myelin content is less, and the axonal distribution is just completely different. I want to point out, because those are the two big groups called white and gray matter. And people that work with myelin staining usually look at the myelin intensity. Maybe they do analysis on the anisotropy in a specific regions of interest. But the question is if we can go beyond that, and we can automatize this classification of brain tissue, not just by myelin content, but taking also into account the tissue arrangement itself. And the method we propose is autoencoders, which is good in feature extraction, or can be used for feature extraction, and it can be also unsupervised, that's what we want. So the main idea is that we have two parts, encoder and decoder. I like to think them as a funnel, because it's just simply as that, the idea. So we can pass the images through the funnel to extract features that are called latent space representation. And then from these features, we can reconstruct again the images. So here the point is we want the latent space representation because it's the features of the tissue. And the quality of this latent space depends on the architecture, that is the funnel. It depends on the dimensionality of this latent space, as well as the amount of data that we use, because this is machine learning, so we need to train the machine. The data that we use comes from 10 rat brains, 5 sham operated, and 5 after mild traumatic brain injury. This makes a total of 500 sections, which is a bit more than 500 gigabytes. And this is how the sections look like across the brain. Here notice that the sections are not consecutive. We stain for myelin 1 every 5. And of course we need some preprocessing. Briefly, we convert to grayscale because we just need the intensity. And then we find that some sections are brighter than other sections, even in the same brain. And this is not because of the tissue is different, it's because of the staining process. So we need to correct for that. The method that worked better for us was histogram matching. So we pick a reference section per animal, and we use this reference to correct the other ones. We did the correction not just per animal, but also among animals. And finally, that is what Christian was just saying, we divide the images into patches, into small squares. I went for a small window sizes, 64 by 64 pixels, which is 14 micrometers. And those squares of tissue are the ones we use to train the autoencoder. So those squares are our input, that we will extract 128 features from them. Okay, but how we extract? The funnel is not a funnel. It's filters, like several filters in each layer that allow to extract these features. And in the last step, we have a dense layer to reach the 128 dimensionality that we are asking. I think there is not really much, well, MSE loss, Adam optimization, and 512 batch size to train. Okay, and with that, we train with 400,000 squares from 50 sections from 10 different animals. This is around 200 million squares. And as I said, what we really are interested is in the latent space. So we, after training, we just pick the encoder. And we pass all the squares of tissue through the encoder to get the latent space representation. And now that we have these features, we can go to cluster them. We use Gaussian mixture models. And when clustering, it comes the question of how many clusters we use or we pick. When checking for the optimal number of clusters, what we find basically is the more the better. I try to cluster data from individual animals as well, like those are the top rows like in color, as well as combining data from different animals, but there is not really a clear optimal. So today I will show you results for 10 clusters. Here we have again histological rat brain section, stained for myelin, and now we send every square of tissue to the to the cluster, and we get this colorful map in which clusters 7, 8, 9, that is more purple, represents areas that are highly myelinated. Then we have clusters 2 and 3 in blue that are the borders. And then we have 4, 5, 6 that are kind of regions in between. Good thing, we can apply this method to as many sections as we wish without really needing time. And now, if I show you this section, if you're familiar with rat brain and with my TBI, you might notice that there is something happening. But if I put the clustering, it's more obvious that both sides are not identical. Here I should say that traumatic brain injury goes across whole brain, but we can see that in the left side, there is more damage. To make the comparison better, I will put sham and mild TBI side by side. So here we can see, for example, the cortex of the sham has myelin presence with cluster 7 and 8 or more myelin presence, but the mild TBI has lost this cluster, so it went to 5, 6 cluster. If we look at the external capsule, I think this area is really evident that the sham is predominating by cluster 9 and 8, but mild TBI went to 7, so we really have this loss of myelin. And now, if we go closer to the hippocampus has is a quite delicate area that with mild TBI is really affected. And if we look hippocampus of sham, we can see clear layers that they are not that clear in mild TBI. For example, stratum radiatum in sham is mostly cluster 4. In mild TBI is a mix of different clusters. If we look granular cell layer is in sham is cluster 2, but mild TBI goes to 1 and 0, really telling us that we lost myelin. This is visible in the histological images. I don't say that, but I think the clustering really remarks this situation. So with all this, we go back to the question that was if we can automatically classify brain tissue properties. We pick histological sections, stain for myelin, we train an autoencoder to extract features, and we cluster those features, and we get a meaningful map clustering. So I would say yes. Of course, there is still a lot of work to do. Right now I'm working with different patch sizes or different window sizes, as well as more or less compression, like what would be the optimal compression. Another thing I have in mind is what's healthy and what is diseased tissue. And in this image, there are some dots. Here, I just illustrate some, but it's full of dots. Those dots is axonal damage. And right now, I'm not able to cluster this damage, but I think it's doable. So I would really want to focus on this as well. And as also adding different modalities. I said that we have 1 every 5 stained for myelin, but we also have Nissl staining for the same brains. So this method can be applied as well into NISL staining. One thing to do as well is to co-register and merge the information from those two different stainings. With all this, I would like to acknowledge the funding resources, as well as thank you all the colleagues from both groups I'm into, and I would like to hear your thoughts. Thank you.